Welcome, everybody. It's really lovely to have you joining us tonight. I'm Kate Phillips, one of the coordinators of the Museum Victoria Lecture Series, where we delve into the museum's collections and research. And today we're taking a voyage of discovery to the deep sea on board the Investigator research vessel, studying life on seamounts in Australia's Indian Ocean territories. On behalf of Museums Victoria, I'd like to begin by respectfully acknowledging the traditional owners of the lands where we work, the Wurundjeri and Boonwurrung peoples of the Eastern Kulin Nation, and First Peoples language groups and communities across Victoria and Australia. We pay our respects to their elders, past, present, and their emerging leaders. The theme of the talk for today was inspired by the extraordinary objects in the exhibition Treasures of the Natural World from the Natural History Museum in London. The exhibition features objects and observations from the voyage on the sailing ship, the Challenger, which set out in 1872 and spent 4 years circumnavigating the globe, observing and collecting and making many discoveries, laying the basis for modern understanding of the oceans. But back to the recent voyage. Tonight, our first speaker is Dr. Tim O'Hara. He's the Senior Curator of Marine Invertebrates. Tim was the Chief Scientist on the Investigator voyage in July. He's a marine biologist who uses museum collections to answer large-scale questions about the distribution of seafloor animals and their evolution. His specialty is the ophiuroids, the Brittle Stars, a class of echinoderms that are an important part of seafloor fauna. Today, he will explain the aim of the voyage and how this was carried out in practice and what was found despite a few challenges along the way. So, over to you, Tim.
Thank you, Kate. Appreciate that. Okay, here we are. So we're going to go off on the Investigator. As Kate was saying, we were very fortunate enough to get some ship time on Australia's magnificent marine open water vessel, the Investigator, the RV Investigator, which is managed by the Marine National Facility on behalf of the CSIRO. And ship time is very special because it's quite a competitive thing to do to get ship time on the Investigator. And what we're planning to do is go and discover seafloor life in the Christmas and Coconut Island territories of Northwest Australia, which are really unexplored, as you'll see in my talk. So it's an exciting thing to do. It's an exciting thing that the CSIRO and the government sort of saw fit to actually sponsor our trip. And what we've done is we drew investigators and researchers from all around Australia. And in all, we had a crew of about 53 finally in the end, and we took off. But you'll see the story isn't exactly as we planned. So why do we do these sorts of things? Well, it's really is a voyage of exploration. I mean, we see ourselves as very much in the tradition of the Challenger, as Kate was saying before, you know, the early historic ships that went across the oceans to discover life on islands and on the seafloor. You know, there's 70% of the globe is ocean and 50% of the globe is deep ocean. I mean, we're not only the blue planet, we're actually the deep blue planet. And a lot of that is totally unexplored. So we can go to places that haven't been mapped. We can go to places that we've never taken a sample. We don't actually know what lives there. So they're kind of really special places to go. So we see ourselves very much as still continuing that tradition of going there, finding out what's happening, and that will lead to definite conservation outcomes. So the Investigator, it's operated, as I said, by CSIRO at the Marine National Facility. It's a big ship. This is not a small little thing. It's 97 meters long, 9 stories high. It can take up to 60, including 20 crew. In our voyage, we had 11 CSIRO technicians, 22 scientists and communicators as well. And as I said, from a variety of places. I mean, we had 7 people from Museums Victoria, but also 5 people, scientists from the Australian Museum, 4 from CSIRO, particularly the Fish Department down in Hobart, 2 from the WA museums. We had 3 universities represented: James Cook in Queensland, the University of the Sunshine Coast, and the University of Tasmania. So a geologist came along as well. And we had 1 person from Bush Blitz who was communicating constantly with different schools and sending out various social media. So it was a fantastic team. But it's no mean feat again, just to try and put everything together. I mean, this took months of preparation. We had to put together 9 pallets worth of jars, bags, microscopes, and gear, and we were carrying over 1,000 litres of ethanol that we used to preserve specimens. So it's a big deal. So what we were planning 45 days at sea. We were going to leave from Darwin. Hope you can see my mouse here. And then we were going to go down northern Australia into some of the really remote marine parks that we have. This is called the Argo Rowley Terrace National Marine Park. And then across what's called the Roo Rise up into the Christmas Island territory. So we're really sort of hugging the Indonesian border just here, and then going to Christmas Island. Then we plan to go to Cocos Island territory after that, and then head back to Fremantle. And it was going to take, as I said, 45 days. This is a major project, $6 million. We were planning to take 100 samples in all sorts of depth ranges through those areas. Collectively, those 2 areas is as large as New South Wales. So it's really an immense area. And we were going to take underwater video, we were going to map, we were going to take a lot of oceanographic measurements, and we were going to try and spread the word as much as we can through social media and media. And why go there? I mean, they're a long way away, but that's part of the attraction, because it's really a voyage to the unknown, as I said before. And these places are pretty special. For a start, there's lots of sea mountains in that region. So these are vast ancient volcanoes that rise from the seafloor, sometimes 4 to 5 kilometers high, which is enormous. That's twice as high as any mountain in terrestrial Australia. So really amazing things. And these have been around since the dinosaur eras. And some of them haven't even been mapped. We just had no idea how big or how dramatic they were. It's also a very interesting area because it's sort of, even though it's in the northeast Indian Ocean, in fact, we expected to have a mixture of animals from the Pacific and the Indian Ocean, as I'll show you later. So it's actually a very exciting place to be, a very dynamic biological place. And the other reason is that the Australian government have announced 2 massive new marine parks for the region as well. And so our work will go directly into helping them prepare management plans for those areas. So that was the plan. We were just going to hop on the ship and we were all going to meet in Darwin and take off. But of course, this being 2021, and who hasn't had a holiday or a trip destroyed over the last 2 years? And in a way, this came very close to being the same thing. So Melbourne was under lockdown 5 just before we went. And so all the Victorians elected to go to the Howard Springs quarantine facility for 2 weeks. And that was an amazing experience in itself. So you can see some pictures down here. It's kind of like a holiday camp, really, where there's no alcohol. So you're locked in there for 2 weeks, and there's quite a lot of other people in buildings around you. There's a bit of movement. In fact, we were in there with a school group, so that was really interesting. A school group of Indigenous kids who go to school in Melbourne, who are going back to the school holidays. So that really livened things up as well. So it was a unique experience, and enabled us to prepare for the voyage as well. So we were working non-stop when we were there as well. But it wasn't just us, then New South Wales went into lockdown. And so all the researchers coming from New South Wales had to flee, New South Wales at one point, to flying to Adelaide or flying straight to Darwin a week or so early. And then just as we were about to go on the ship, there was a lockdown in Darwin. I mean, Darwin hasn't had a lockdown before, but this just happened right before. So even though we'd assembled everyone in Darwin, we suddenly had to bolt for the ship to make sure we got there before any impending lockdown and get into a COVID-safe place. Once we're on the ship, we quarantined for a couple of days in port, and then we set off. So I called it the COVID Great Escape, and it was really like that. It was just nerve-wracking, exciting in some ways, but finally we assembled. We'd lost 2 people on the way who couldn't escape the COVID situation or whatever, but we assembled most of the people and we were ready to take off, and we did. So it's a long way from Darwin to Christmas Island. It took us 6 days, in fact. And most of the time, we're just looking at sea. It's really interesting, as I'll show you in the next slide, some of the things you can see from the boat. But all the time, we were mapping as we were going. And the underwater seascape was pretty amazing as well. And this is just one feature I want to show you. This is way near the Australian border in the Argo Rowley Marine Park. And it is an enormous canyon, which these pictures almost don't do it justice. So this is our track here. This is what we can map underneath the boat using our multi-beam sonar. And this is an enormous canyon that cuts into the edge of the Australian continental plate. These are enormous things. They're not actually currents of water, they're currents of sediment. It's just like a sandy beach. When the waves start eating the sand away, they kind of sometimes the sand just drifts away in rivulets. It's very much like that, but on the seafloor. So you get these enormous kind of features, really interesting seafloors. And ocean life is, of course, always interesting. We did see some blue whales and some spinner dolphins, but weren't fast enough to photograph them. But we certainly saw a lot of flying fish as well. These are magnificent creatures that can go, almost 100 meters or more flying in the air. The boat would disturb them and they would shoot off in front of the ship in various directions. And this brought the birds, particularly as we got nearer and nearer to Christmas Island. We had loads of boobies. There's 4 species of boobies that live or nest on Christmas Island, as well as trophic birds over here. And these things here, these are kind of the Death Star or the Death Ray, I don't know, the death flying ships of the oceans. They hover, they're frigate birds. They hover way up in the sky, and if they see a booby's got a flying fish, they dive and start pulling on the tail feathers to make them drop the fish. So it's all very dramatic. So this is what happened. So here we are over here. So there's our voyage tracks. You can see how much we're mapping as we go. And the first thing we did, we hugged the boundary because we were aiming for a really deep spot where the seafloor was supposedly down to 6,500 meters. Then we're going to Christmas Island. Then we're going to head to these seamounts, explore these seamounts down here, as well as there, these ones over here. And I'll show you maps of all these places. So this is a graph. I know a lot of people don't like graphs, but if I'll explain it to you, hopefully you'll see what I'm talking about and you'll get the importance of this. So this is the track, as I was showing in the last map. And if you can imagine you're going along here, you can see that these are a deeper bit and then you're going over a ridge. And this is what I'm trying to represent here, because this is depth on this side. This goes from 5,000 meters below the sea levels, that's 5 kilometers down, to 6.5 kilometers down to the bottom of this graph. And the red line is what we mapped. And the green and blue lines are what people used to think was there from the gravity maps that you can get from satellites. So we know what the seafloor looks like broadly because you get these fuzzy kind of gravity readings. So you can see that this is probably a bit of a rise, maybe a seamount. These bits are really deep. So we're expecting really deep seafloor, and it didn't pan out that way. We're expecting these really deep holes. It more or less was kind of flat at 6,000 meters with these small ridges going across. So that was a real surprise. So we didn't find the deep holes we were expecting, but we sort of found a really deep plain. This is some of the deepest seafloor in Australia. Then we arrived in Christmas Island. So here's the Investigator. Someone had a drone ashore and actually flew around the ship, and we managed to get the footage. So we got the magnificent ship against Christmas Island here. And this is what we did. We went around twice, we mapped, and then we started sampling all these different locations. And we could produce these beautiful maps of exactly what it looks like. And Christmas Island is a seamount as well. It's just one that comes above the surface. So this is a kind of a profile here. So this is at the edge of it, and then we're going to go up the slope of the sea floor here up into the island and then go down the other side, and that's what you can see in this profile. So it's only that little bit at the top above zero, so that's sea level. That's actually the island, but it actually sits on this enormous seamount underneath. It's just phenomenal. Again, you think Kosciuszko is only 2 kilometers high, so it's only going to be up to about there. And we've got all of this. It's a massive structure that sits below the sea level, and you'd never know from the surface. So that was exciting, and it was exciting because it actually does go so shallow. So we could actually have a look at the animals that were sort of between 400 and about a kilometre deep, 400 metres and a kilometre deep. And the next talk after this, Melanie will actually, she'll show you most of the animals. I'm just going to show you the maps and where we went and the reasons we did it, and Mel will show you all the beautiful creatures. So after that, we headed off to explore the seamounts. So these would have been above sea level at one stage, hundreds of millions of years ago. So this seamount started, the oldest rocks are from 107 million years right down the base. So it would have been a massive ancient volcano, and it actually came above sea level at one point, and the waves have made it flat on the top like that. But then because the lava is so heavy and so dense, it actually sinks into the soft oceanic crust. So over time, these things rise up with all the larvae, but then they sink again. And so this is now down to 2 kilometers below sea level again. So you've got this whole life cycle of seamounts, which is pretty amazing. So they're ancient structures. And in fact, you can even find fossils from the dinosaur era on top of this seamount. People have, of shallow water corals and other things. So yeah, pretty interesting. So we surveyed the top. The top tends to be now covered in deep mud or sand. But on the sides, you tend to get a lot of rocks. So you get sponges and corals and other things growing around. After we saw that seamount, we headed south, and as we were having dinner one night, suddenly we went over this massive caldera that no one knew was there. Right? It was just a fuzzy blob on the gravity maps, and we just revealed it in all this beauty. It's just amazing. This structure, as you can see from this profile here, it's 20 kilometers across at its base. So this is an enormous volcano that's the top of it's collapsed. All the hot lava has been thrown out of the volcano. It's left empty chambers and the top collapses. So you get this caldera forming. And who knows when this happened? This is, again, could be hundreds of millions of years ago, or it could just be recent. But it's a really amazing structure, and it really excited everyone on the ship, not just the geologists, but everyone, all the biologists as well, and the crew. It was just amazing to see. And so we nicknamed it the Eye of Sauron after the Lord of the Rings books. And this became the Ash Mountains, which was very pertinent because it's actually topped by a lot of pumice stones, in fact. And the pumice stones, we believe, I haven't proved it yet, actually came from Krakatoa seamount that blew a couple of hundred years ago. So then we headed to another set of seamounts called the golden bosunbird seamounts. Golden bosunbird, one of those birds that I showed you before. That's its common name on the islands, but someone poetically named these seamounts after that. But these are also enormous. They go up to about 1,000 meters below sea level. So they're much shallower than that one I showed you before. So again, very interesting, the different sort of animal life that you see on the tops of these. Some of them have definitely been above sea level before because they've been planed off like that flat shape. But others are still really rugged, and so perhaps they never got above the sea level. They've always remained underwater mountains. So yeah, lots of really interesting places. So again, these are our sample sites that we took. So we tried to actually get a range of depths to understand where the animals, how the animals live there and where they live and whether they're living at different depth ranges. I'll show you later. But once we got to the end of Balthazar, unfortunately, there's some equipment issues, and the ship had to go back into port. So we never got to the Cocos Islands this time. Hopefully we will shortly, as I'll explain later. So we began the early and very long transit home. The idea was to actually, we left those golden seamounts and we headed south, and the idea was to go to Perth. But again, COVID played its tricks, and at that stage there was another ship in Fremantle that had a lot of COVID cases, and the state government was quite nervous about our ship coming in. We may have got in, we weren't sure. And so in the end, the decision was made that we wouldn't dock in Fremantle. In fact, we would go all the way back to the ship's home port in Hobart. So it became this epic return journey. We basically semi-navigated Australia almost. or halfway. And it was eventful as well, because we ran into two really large storms. There was a large storm off WA that had 9-meter waves, and another one off Tasmania that we just managed to outrun. So it was an exciting trip back, a bit bumpy. And it was very nice to see Hobart. When you come in up the Derwent, this is its home port. You come in, there's the centre of Hobart there, and you get a bit closer, and then you see CSIRO, and then you dock. So it's an exciting time. It was great to be back. So I'm going to tell you about one group of animals, because of course we've been in lockdown ever since we've been back, and we haven't actually managed to distribute the animals to the other museums as yet. We've been prevented from doing that. That will happen as soon as we come out of lockdown. But for the time being, I have actually managed to identify one group of animals. So these are the Brittle Stars, and these, as Kate was saying before, these are my favourites. And so you can see all these jars here, and each jar has a different species for a different location. So there ended up being 46 different species of these animals across Christmas Island and all those seamounts. And some of them are beautiful, actually. Or this is a hunter. This is one that likes to eat kind of dead fish or other carcasses on the seafloor. These are quite large, maybe 3 centimeters across the disc there. And other ones kind of like snake-like, almost. They have 5 snake-like arms that grip onto things like corals and feed off the plankton. So they're pretty exciting. But what I was using them for is to try and work out, do the animals differ with depth? Do they differ on different seamounts? And so I've just only started doing that. We haven't been back long. But what I can tell you right now is definitely they change with depth. I can see different groups of animals at all of these depths. So you get this abyssal fauna that is chilled to 1 degree Celsius. That water comes all the way from the Antarctic. And then you get another set of animals that live between 1,900 and 3,300 meters. And that's the Indian, they live in the Indian deep water, which is about 2 degrees. So another set of animals that have adapted to that range of temperatures and depths. And then there's another set of animals that live in what's called Antarctic intermediate water, which comes all the way from the subantarctic, sinks below the ocean and gradually moves north. And the upper layers are bathed in what's known as the Indonesian through-flow currents. And this is an enormous current that comes from the Pacific, goes all the way through Indonesia, and then sweeps out across the Indian Ocean. And Christmas Island is just about there. And so it actually is right in the center of this current. And it carries a lot of animals all the way from the Pacific all the way through Indonesia to Christmas Island, which is why I was saying it's such an exciting place, because you actually do get some minor currents, say, bringing stuff down from the northwest Indian Ocean, but you get this major current coming all the way from the Pacific, so it's a mixing of faunas. So depth and currents definitely matter. That's as far as I can tell you at the moment. But what we have been doing is identifying key areas for diversity for the Parks Australia, who are going to manage these new parks. And we've identified all these special habitats. So we talked about the really deep areas. So it's more of a plain than these holes, but it's certainly one of the deepest areas in Australia. There are enormous Christmas Island seamount that comes way above the surface and forms an island. You've got those golden bosunbird seamounts here. You've got a nodule field. And when we go back there, this is what we hope to find in the Cocos. We hope to investigate this ridge system. It's called the Investigator Ridge. Cocos Keeling Islands sit on another enormous seamount. And there's another one here that almost comes to the surface. It's actually only 16 meters below sea level, as well as some of these deeper ones. So, there's lots of exciting things to come. So these are the new marine parks that have been proposed by the government. And in fact, what they've decided to do is incorporate almost the entire area of the territories in both Christmas Island and around Cocos Keeling Islands as marine parks. So this is one of the most significant marine parks in Australia, in fact, worldwide, really. Between those two areas are larger than New South Wales. They're larger than the country of France. So it's extraordinarily large marine parks that will help protect biodiversity, and in particular endangered or vulnerable marine species like tuna. It's twice as large as the Great Barrier Reef. So they're only proposed at the moment, and there's the community consultation process, but with any luck, they'll be proclaimed next year. So what's next? So we're going to continue to identify the treasures from IoT Voyage 1, as Mel will show you soon. And we're going to start already planning for the next voyage. We don't know exactly when it will be as yet, but still these things take a long time to plan. So we've already started doing that. And we'll have those new marine park proclamations. Thank you very much for listening. And now to you, Mel.
Thank you, Tim. That was absolutely fascinating, and particularly liked the fact that you ended on that bit about the marine parks, the potential new marine parks and how big they are. That's quite amazing, really, and very heartening in terms of their role in conservation. Our next speaker is Melanie Mackenzie. She's the collection manager of marine invertebrates, and she's been involved in several major voyages of shipboard research, from going to the Australian abyss to Antarctica. And she has special interest in holuthoroids. Sorry, that's a hard word to say. Sea cucumbers is easier. Mel will show us a typical day at sea, from specimen collection and processing to the practicalities of eating, relaxing, and sleeping on board. She will also introduce us to a range of the amazing fish and invertebrates collected by the team. Over to you, Mel.
Thanks, Kate, for that lovely introduction, and thank you all for joining us tonight. So as Kate said, my name is Mel Mackenzie, and I'm a collection manager of marine invertebrates at Melbourne Museum. So when we're not in lockdown, I get to help manage this amazing life library of specimens that's accessed by researchers from all around the world. And I also have a soft spot for holothurids, sea cucumbers, like this very cute one here with little babies on its back. And these sea cucumbers have taken me to some amazing places around the world, and in particular on a lot of voyages, like the one that Tim just spoke about. So following on from Tim's talk, I'm going to give you a little bit more about life in the floating office and what a typical day was like on board, and then also give you a sneak peek at some of the animals that we saw. First, a quick tour of the Investigator. So Tim gave you all the sort of facts and figures, but just so you know, these are the kind of areas that we were mainly working in. So we had the workspace at the back there, the aft deck and the labs. Monkey Island at the top is when we had some time off and you might want to look out and see some whales or something. The bridge was where the captain or the master was controlling the ship from. The scientist cabins, you can see we had some up on level 3 and some down below the waterline. And there's also, of course, the bow where we could watch some animals and get some fresh air. So this is the bridge. Looks a little bit Star Trek-y. This is where the master would control the ship from. And there's that space at the front where we could get some fresh air, watch some beautiful sunrises and sunsets if we were lucky, and maybe even spot a booby or some whales. There's the aft deck there. So that's where all the machinery was, the equipment was deployed from. And we were lucky with invertebrates. You can see they were doing a lab induction. So we had this wet lab where the material would first be processed. And then we also had a dry lab where you could go to work on the material again later once it had been processed. And fishers also had a lab, so they did all of their work there. And this is the operations room where Tim spent quite a lot of his time doing all the mapping and things. And we had some recreational lounges and a mess. So you can see there one of the lounge at the top, people doing some jigsaws or reading books or watching movies on their computer. There's a science talk going on in the other lounge there. And you can sort of see Jeremy in the corner there sitting in a racing car game. And we also had things like ping-pong tables and Kate sitting there in the galley where we'd, of course, eat our meals. We also had some gyms on board, 2 gyms, because of all the COVID restrictions. So you could only have one person in at a time. And you can see there the equipment that we had was really quite heavy equipment, but also it's all really lashed down, tied to the floor. So it couldn't possibly move, but you could fall off it if it got really rough. So yeah, I think it was really on calm days that the gym was used more often. But in some high seas, it can be quite fun to use that rowing machine. And these are our cabins. So most people had a share cabin. You can see the bunks there. Oh, that deadly ladder. I think I nearly fell down that one a couple of times. But, you know, quite spacious really for a workspace, and, you know, quite comfortable. And we had things like block-out blinds. All the drawers and everything could be secured in heavy seas. Even the chairs could be tied to the floor. A lot of storage space. And you can see there in the shower, you have things like handrails everywhere. And the beds themselves, if you're sharing with someone else, it's nice to be able to pull a curtain across. So you've got your own space and you can keep the light out as well. The best thing about living and working in a floating office is, of course, the changing view, which we really miss being in lockdown at the moment. But you can see there, Katrina up in the whale watching, the Monkey Island, the whale watching area. So she would have had a great view like this at the front. So now just a little bit about life on board. As Tim said, we had a very strange start with that dash to the ship, the scooter race. But we also arrived to a ship quarantine, of course. So we all got taken individually to cabins and were told to stay in our individual cabins by ourselves. So it's a little bit disconcerting for people who hadn't been through quarantine or hadn't been in lockdowns before. But for me, I just got to play with my show bag. So Sarah prepared us quite well. They gave us this little bag of goodies to keep us nicely distracted while they were sorting things out. So you can see there even a stress whale, which was quite handy. And we had 3 pre-departure COVID tests. So there's Rob getting one of them. And of course, we had to adhere to things like cohorts and distancing, particularly in the first half of the voyage. And we also had masks, and we had to do daily temperature checks. Originally, we did individual ones in our rooms. We weren't allowed to leave our room until we submitted that daily temperature check. But as the cruise moved on and we were getting safer and safer, we were able to do the therminator instead, get our temperature taken in the lounge. And of course, as is the case with any ship voyage, we had a lot of safety drills too. You can see Tiff there donning the good old immersion suit, which makes you look a little bit like Gumby, but you can see it's got the joined-on gloves and everything. So if you were to fall into the water with that, it floats quite well. Of course, keeps you a bit warmer for a little bit longer. And with all that downtime, so we did work 12-hour shifts, but yeah, there is downtime when you're not actually working. And of course, we had things like that ping pong table that I'm sure you saw before. And there's a dartboard there in the background, you can see. We had a very good competition going with singles and doubles for ping pong, and Nish was a bit of a star, but I think Tim held his own. And Rob there is doing a tour of the engine room. So it's always great on a ship to see the mechanics that you don't normally get to see. Lena there giving a science talk on worms, and Thomas giving a talk as well. So lots of different things to do during downtime. And of course, plenty of interesting food on a ship. We always had so much food. It was never really a problem with that kind of thing. Though, of course, it's things like salads and vegetables and that go first. So a little bit harder for vegetarians and harder if you're trying to watch your waistline. But there were some very intriguing things that people were eating. I saw this wonderful combination of baked beans and lychees. I don't know who came up with that one. And of course, there was actually an ice cream fridge, which kept everyone happy. And we had lots of birthdays on board too. Tim had his big one, his big 60th. And Alice was amazing and fashioned this Brittle Star, this ophiuroid pinata with lollies in it. So yeah, lots of fun and games. And Pete there with a sea shanty pirate patch on, ophiuroid Brittle Star again, and lots of cakes. But there were actually quite a lot of birthdays on board. I think there might have been 5 or 6 of them. So yeah, that's always nice, break up the time a bit. And of course, when it was a little bit rougher, we'd probably just stick to movies and naps. So if there were rocky seas, you can see everyone there is sitting as low as they possibly can. That's also because those chairs were flying across the room sometimes. And you can see under Anna's legs there, there's some ginger chews. So yeah, I think it's sometimes the stomachs just needed settling and it was too hard to concentrate on anything. And as Tim said, our voyage didn't quite go to plan. So you can see there, the blue line was where we were supposed to go from Darwin to Christmas and Cocos and then back into Fremantle. And the yellow line's where we actually went. So when we turned that corner and got into the southern seas, you can imagine it got a little bit lumpy. And you can see there from the angle that Nish and Claire are on, it was really quite rocky. But luckily we had stabilizers in the ship, so we kind of knew that we shouldn't tip over, but, you know, it still gets a little bit scary. And things are flying around everywhere, of course, if they're not tied down or put down. And of course, everyone was sort of hitting the seasickness tablets, or for me, I usually just stick to ginger. That does a good job for me. So just to give you a bit of an idea of that, the student Alice on board, actually, she was really quite seasick for most of the time, and she wrote down something about how she felt. So I think I'm going to read that out to you while I give you just a gentle example, so you can watch this gentle example of what it was like. So this is from Alice. Fortunately, few of us have been afflicted by seasickness. For those who've fallen victim, it is a poison, a noxious toxin that catches you off guard, that stirs when you step into a human room or is roused when you round a sharp corner. Once activated, there is no antidote, no aid, no escape. No matter how much icy water you splash on your face or how thoroughly you empty your stomach, you are helpless. You can only sit powerlessly and wait as the deadly venom coils slowly through your veins. It methodically infects your vision, your taste, your hearing, poisoning all your senses until finally there is no concept of time and place, no sun, no sky, no sea, until you smell and breathe and dream only despair. Very dramatic, but anyone who's ever had seasickness would know that that's actually what it feels like. And of course, when there's rough weather, we do have to make extra modifications. So things like in the galley, the chairs would have been changed to the floor. We would have put sticky mats down everywhere. We lost a few coffee machines, which was very sad. Things just kept flying everywhere. And in our own cabins, we tried lots of different methods. For me, this was the best method. I did try sleeping on my yoga mat on the floor once, but I fell out of my socks. We moved so much. So I thought, no, that's not going to work. So you can see here that I put the immersion suit under the corner of my mattress to prop myself up, and an extra blanket against the wall. So I'm really just wedged in there. And of course, it's not all fun and games. We do actually have to have a normal working day. So how exactly do we sample below the waves? Well, we can't scuba dive, so we have to use these frames and winches to deploy our equipment instead. This is some of our equipment here. This is a deep tow camera. It's quite beautiful, isn't it? You've got lasers on there as well so that you can sort of measure animals and all the rest, but this gets deployed so that you can see what the animals look like down there and also their habitat, which is great. It's quite specialised equipment, so you can't really put it down in rocky areas, but you can see that Tim was very excited about the first deployment. He was waiting behind that guardrail. This is the CTD. We use this one to basically to collect water. So those bottles, they're quite tall, and those bottles are actually fired off at different depths, and they collect water from that depth so that they can be measured for salinity and temperature and things like that, and also so that that water can be tested for things like eDNA and used for experiments and things too. This is the Sherman Sled. So you can see it's a bit of a chonky one. And you can see at the front, it's got lots of dents in it, so it's obviously hit quite a few rocks. But that's the mouth there, and that's the area that collects the animals as it gets dragged along the floor of the ocean, and it has the nets behind. So the nets trail out behind, and they're tied at the end. They've got a cod end at the end. And basically what you're doing is just doing scientific fishing. So it's a very short, sweet, quick check of what's under there. This is the beam trawl. It's a slightly bigger one. So if we don't catch much with the Sherman, then we might put down one of these to see if we can cover a little bit more ground. And it's the workhorse. Again, it has a cod end at the base. And so you're sort of collecting the animals in the mouth, and they're getting filtered down to the bottom. But even that one can hit rocks now and then. You can see it's got a very bent beam there. Luckily, we had some amazing people on board who are very good at mending nets and fixing beams. And as Tim explained, we of course use sonar to map the deep too. So we didn't know before this voyage what was under there. So we had to actually map before we could put any of that equipment down. And then Tim could decide where was best to land, so where he wanted to put that equipment. In a typical workday, we had the 12-hour shifts. So for the invertebrate team and also for the fishers, but for example, we had the 2 a.m. start, which was our team at the top there, and the 2 p.m. start, which is the dinner and sunsets versus the bacon and sunrise. So I had to quit bacon after, I think, the first couple of days because it's just too much. But first things first, so you get up and you can actually on your phone now using the Grafana app, you can actually check what's happening, like what equipment's been deployed or how far it is from being on the deck. But if you're not going to check your phone, there's computers all around the place that have this up on it too, or you can check the board, the whiteboard. And the whiteboard is actually really the go-to that tells you what operations are happening, what kind of gear, and when it's likely to be landing back up on deck. And of course, another first things first is the lab handovers. So the lab custodians would talk to each other and explain what had happened on the previous shift, anything they'd suggest that we should do or change, just so that we had that continuity between the two teams. And often we'd start with, if there wasn't something immediately coming up, we'd start with restocking the lab. So bringing all that extra vials and things like that in. And of course, turning on the ice machine because we're working in tropical waters. So we didn't want the specimens to start rotting or anything. And the crew actually deploy and retrieve the equipment. It's not the scientists. So on some of the other voyages I've been on, the scientists have been involved in this and I've had a chance of retrieving this equipment myself. But on the Australian vessels, it's the crew who actually pulls in the nets. And they're just really worried about the scientists' safety. So we stay behind the cordoned off area. In this science shelter here, and you can tell they don't trust us. They've put up that little cordon bit so that we don't get too excited and rush out to see what the animals are. We have to wait for them to get the all clear first, and you see everyone there with their safety equipment on, looking very excited about what might possibly come up. And then we check the nets. And you can see there we get lovely little goodies. So we have to sort out things like the fishes from the invertebrates, from the rocks. And then we ferry those specimens back to the lab. And if it's been a muddy haul, then we'd actually have to run them through sieves to get rid of the mud. Or otherwise, we just start processing them on the table. And we normally do this by starting with sorting like with like on ice. So if you see something that looks like a crab, you put it with the other things that look like a crab, something that looks like a jelly with the other things that look like a jelly, sorting like that at first. And then gradually working down into better IDs using photos or literature or all the experts we had on board in different areas who might be able to tell you a specific name for an animal. Experts like Lena for worms here or Tim for ophiuroids there, working through the microscopes. And really important to collect all of the data, so the collection data about exactly what depth it was at and where it was, and also to label the specimens as well. And of course, photography is really important too. So these are deep sea animals, you know, often people would never see them other than in jars in a museum. So it's fantastic to get their live colour or their as near as possible to live colour and to see what they look like before they're preserved in ethanol. And also before they're preserved, taking things like tissue samples or measuring them if necessary. And we had Steph, the geologist, on board, so you can see she's very excited about her rock there. A lot of the rocks had to be cut. We also had some awesome pumice, I think maybe from even Krakatoa on board, so that was pretty cool. And Claire there showing, showing the joys of preserving specimens. So, of course, once you've gone through the process of processing the specimens and photographing them and everything, they need to be pickled. So they need to be put into ethanol or formalin to preserve those specimens for the future. So in museum collections around Australia, for the next 100 years. And when we weren't doing the biological sampling, we were helping with the water sampling with CTP. And to be honest, it actually wasn't too bad. It was quite nice in the tropical waters, accidentally spilling this bottom water on you. It was quite nice and chilly. And we also, of course, helped out with digital stories that Rob's doing there, media and education. So Kate there from Bush Blitz is setting Steph up for an interview about her rocks. And Kate was doing a lot of live crosses every day with different school groups, either on Christmas Island or in Australia. And it was fantastic just to talk to all these kids about science. Lots of really good questions out there. And you can see Rob taking photos of the polystyrene cups there. So polystyrene cups, they're filled with air pockets. And so we would send, you've probably seen it before, scientists sending these down attached to that CTD in stockings, in fact. And we send it down to the bottom of the ocean. And because of the huge pressures at those depths, all those air pockets just compress and these cups just shrink. So if you've drawn a cute picture on there, it gets even cuter and smaller. And of course, we then just do it all again. So it just depends on how many deployments you have in a day. But if there's time, if there's not too many deployments, then you might have time to do some further IDing in the lab. So what do we actually find below the waves? Well, I'll give you a sneak peek. It's not everything, but some of the animals. Lots of very cool critters. Prawn, shrimps, and other crustaceans. Cute kefs, like this Dumbo octopus here. And sea spiders like this one. So not like a land spider at all. These sea spiders are gorgeous and they've got straws for mouths. And squat lobsters with their funny butts and their long arms. And snake stars. So you can see the snake star here as being gripping this coral. And that's actually a really good example of something in the deep sea. Often something like a coral, which would be attached to the substrate, is perfect habitat for other animals to hold on to. So this snake star would have been holding on and reaching out and feeding from the water column, which you know, it got to prop itself up a little bit higher by being able to hold on to that coral. And jellies like this one, urchins, brittle stars, Tim's favourites. These guys are everywhere and they are very brittle. Then holothurios, lots of sea cucumbers, which I love to see, my favourites. Sea stars, corals and corallimorphs. Line lobster, this is another really good example of a deep sea animal. You can tell because it's red. So of course, red's the first colour to be lost at depth. So really in the deep sea, this animal would look black or invisible. So that's quite a sneaky way to be. So you'll find a lot of deep sea animals are actually red. And also this one's also lost its eyes because it doesn't need them down there. Goose barnacles, I love goose barnacles. So they're barnacles, which means that they're actually related to crabs and crustaceans, but they look like shells. And it's not until you sort of see them kind of open their valves and put out these little feather hands that you realise that they're more closely related to things like crabs, but they filter feed and everything as well. And they're amazing. They can be quite parasitic. You can see them here attached, I think that's to a bamboo coral there. And there's even a little one there, if you can have a look at the tusk shell, the scaphopod there, and you can see that there's a little barnacle on the side there. So they like to just basically attach themselves to anything they can in the deep sea and move around. And that's a pancake urchin there, or a Scotch bonnet. It normally, when it's under the ocean, it actually fills up with water and can be quite high, but as soon as it comes up, it's flat as. But it's also quite poisonous. So we had to keep warning people to make sure that they had their gloves on or they were using forceps just to not actually touch these animals, because a lot of deep sea animals, we just don't know what toxins they might have, what spines they might have and things like that. Even the fishes looked quite odd, but they did have great names like this tongue sole. And this one you can see that the fins have all merged and the eyes are actually on the adults, they're actually just both on the left side. Giant cusk eels, look at that guy. And tiny batfish, I love the batfish, they were so cute. And you can see they've got that modified fins that they actually use to kind of almost walk across the seafloor. And they've also got a lure. They're an anglerfish, so they have a lure, but it's not like the light bulb lure that you see in cartoons and things. They think it might be a chemical lure. It's quite interesting, yeah. And a conger eel. So these guys have no scales and often have small teeth. And the opposite here, the Sabretooth fish obviously has very large fang teeth. And this is a lantern shark, and it actually has a whole lot of lights underneath. And the dragonfish, these guys are amazing. Look at that. Look at that lower jaw. It almost looks like a pug. And it's got that great light organ on the side. And again, it's got light organs underneath too. And you can see the lights are used in so many different ways in the deep sea, either to get rid of your shadow so that other things can't see you, or to attract prey. And there's thought that this light organ on the face here is potentially a torch to be able to see animals that are red. So, you know, who knows? There's a lot of different adaptations that are still being discovered down there. And this is a tripod fish. So these ones actually sort of prop themselves up on their fins like a big tripod, and they sort of turn into the current so that they can, I guess, to feed. Pretty amazing. And fang-tooth. You'd be really worried about this if you didn't realise how small it was. But check out those teeth, they're huge. And these guys have actually got a sheath in the top of their mouth so that those bottom fangs, can't go up too high. They go into a sheath, so it's like a pocket in the top of the mouth. And the cookie cutter shark, this is one of my favourites. It's actually bioluminescent, which is pretty cool. Again, a lot of deep sea animals are bioluminescent. And this one, look at those teeth. It's almost like a cookie cutter or like an apple corer. It basically just takes chunks out of other animals. So it doesn't usually kill them. It's just taking a bite, a bit of a nibble out of a seal or another shark or something. And everyone's favourite, just because of the name, definitely not because of the looks, was the bony-eared ass fish. Named by a German guy. They think it might have been named after a donkey rather than something else. But anyway, the weird thing about it is it's actually got the smallest brain to body weight ratio of invertebrates. So, and of course, another link again, it was first collected, the first specimen was collected from the Challenger in the late 1800s. And above the waves, we were lucky enough to also see some things. Gorgeous views of Christmas Island, amazing night skies, and lots of really cool visitors. I love these flying fish. They looked tiny from the top of the ship because the ship's so high up, but we had some that landed on deck, and they're actually, much bigger than you think. So, and some curious seals as we pulled into Hobart. And my first blue whale, not that we could really see the whole animal, but, we knew it was there. And lots of birds, amazing. I love the boobies. Just the blue face looks so cool. And the frigate birds are very nasty, but very cool. And lots of beautiful sunrises and sunsets and seascapes. We were just so lucky. So thanks for listening, everyone.
Thank you so much to both of you, to Tim O'Hara and Melanie Mackenzie. It's been great getting a taste of your time at sea and a real sense of the excitement of discovering what's deep, deep down there.